Welcome to the Bible and Science. This is Dave Prentice. I'm a retired public high school teacher, certified in all branches of science and math. I've also taught on the Bible and Science for many years, both in the U.S. and on many overseas mission trips. This is episode 27 of the Bible and Science. It's a continuation of episode 26. We're continuing to talk about the origin of life, whether it could be something that's the result of natural processes only, or whether it requires supernatural intervention. Quick review. In the last session, we talked about the fact that if we're going to produce life by chemical processes, we have to get oxygen out of there. Free oxygen is the second most reactive element there is as far as taking away electrons, second only to fluorine. So we have to make sure that there's no free oxygen because it's going to interfere with the chemical reactions. Number two, if we've gotten rid of the oxygen, here's the other problem. The sun is putting out a bunch of ultraviolet, including the wavelength of 310 nanometers, which is deadly to living things. So if we get rid of the oxygen, we've gotten rid of the ozone layer that filters out the deadly microwave, the deadly 310 nanometer radiation. And then also we need a very sophisticated trapping mechanism that's going to allow chemical process to take place. We get just a little bit of energy at the right time, at the right place, and then we have to do a little chemical stuff, and then we get just a little more, and meanwhile we have to remove the products because they'll be damaged by the same energy source. So there's no such thing in nature. And then proteins are made out of amino acids, which require amine groups, which is an NH2, which means you have to have separated nitrogen atoms. It's called nitrogen fixation. The atmosphere has a lot of nitrogen, about 78%, but nitrogen is so unreactive in its natural state that it can be used as a fire extinguisher. So somehow, in order to produce amino acids, you need to separate the N2, which is two nitrogen atoms bound together into a molecule, and then you can do stuff with it. Well, the problem is that this is not something that would just occur on its own. Now, a great deal of the nitrogen that's fixated is done by bacteria, but the bacteria are living things and they required fixated nitrogen. So the alternative would be if you use lightning. Well, lightning is so strong, it's going to cause an awful lot of damage. Okay, moving on to new problems. Amino acids. There are 20 amino acids used in living things. There's bunches and bunches of other kinds, but the only ones used in living things, there are 20 forms, 19 of which can exist in mirror images known as optical isomers or enantiomers. There's a left-handed form, which is abbreviated L for levorotary, or right-handed D for dextrorotary, of each of these. So they're mirror images, but they're not superimposable. That's enantiomers are non-superimposable. So, for instance, if you look at L-alanine and D-alanine, they're three-dimensional molecules. If you put one on top of the other, they point different directions. So random chemical processes produce a 50-50 mix of left and right-handed amino acids, but you need 100% left-handed amino acids. Oh, and also you need 100% right-handed bases in the DNA. But proteins in living cells are not made of a 50-50 mix. They're made of only the left-handed form, the levorotary form. There's no way you can explain this by chemical processes alone. There has to be something to purify it. So what you do if you're an experimenter doing this kind of research is you go to a chemical supply house and you buy the purified form. In nature, you don't get the purified form. It requires intelligent intervention to separate it. So let's suppose instead of 20 different type of amino acids, there's just one type of amino acid used in cells instead of 20. So could random chemical actions accidentally put together just 125 proteins of 100 amino acids each using only the left-handed form? Well, if you flip coins 125 times in a row, what's the chance that you're going to get all heads or all tails? It's just not going to happen. So this is just about as likely as a groundhog that we referred to last time, with a 50-50 chance of making it across any one lane, getting across 12,500 lanes on a superhighway. Yeah, he might make it across the first, second, third. By the time he's at the 10th lane, he's only got a 1 in 1,000 chance of being alive. By the time he's at the 20th lane, 1 in a million. So he's got to go to the 12,500th lane on the superhighway to reasonably expect that he's still going to be alive. Okay, is it impossible? No. Is it reasonable? Certainly not. So what if evolutionists find something that can't be explained by known natural causes? No problem. They explain it anyway, by making up stories about unknown natural causes. The story we're going to do here is chemistry and time. Aha. So let's talk about chemistry. Remember, at the bare minimum, you need schnapps. I'm leaving out the phosphorus right now just to put together 18 of the 20 amino acids. But you're going to have to start with the right chemicals to make amino acids, bases, and sugars. And then hundreds or thousands of these are going to have to overcome the natural attraction of positive to negative, negative to positive, so they can work their way past all of these useless oppositely charged chemicals and link into each molecule of protein, and along the way RNA or DNA. Now, once we've got a few molecules of protein, DNA, RNA, these are going to have to link themselves together into clusters, which are called coacervates. At least some of them must be able to attract the other molecules they need, because as I said, the majority of what's in your cells is schnapps, but there's a bunch of other stuff besides carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. You need a bunch of other things. So somehow these coacervates are going to have to be able to attract the other molecules that they need in order to put together a cell. Now they need to protect it. They need to form a cell membrane and absorb exactly the necessary molecules to put together a mechanism such as RNA or DNA. RNA is ribonucleic acid. DNA is deoxyribonucleic acid, which is RNA where you've removed one of the oxygens. So somehow the molecules have to come together to put together a mechanism such as RNA or DNA in order to be able to reproduce. And then finally, the whole thing has to become alive. What's that process? We have no idea. Life coming into existence from non-life, we don't have an explanation for it. A cell is more than just a few amino acids strung together. Steve Green had a wonderful song called All Across the Sky, and he said in it, "What about the mystery that cuts us like a knife? What is the source of life?" We don't know. Why is something alive and something else is not alive? They can be chemically identical, and yet one is alive and one is not alive. So these are the 20 amino acids that are used in proteins. Notice 19 of the 20 have either an L or a D. No Ds. Have an L in front of them, meaning they're the left-handed form. The only one that only has one form is glycine, but all of the other ones can exist left or right-handed. And in proteins, in living cells, all we use is left-handed. So let's say we put together a primordial soup. We're going to contain carbon, hydrogen, nitrogen, and oxygen so that you can see the amino acids coming together as on the previous page. Well, even with such a simple primordial soup, the chemistry is so complicated that experimenters will buy purified amino acids instead of making their own. But even then, that soup is far too simple. 2 of the amino acids require sulfur. And then, oh, wait, there's other parts of a cell besides just amino acids, aren't there? Yeah, DNA and RNA both require phosphorus, so throw that in the mix. Early plants, which are supposedly some of the first living things, photosynthetic plants, or something that would be similar to plants, they would require magnesium for photosynthesis, so we're going to have to throw magnesium in there. And then many living things also require such parts as iron, copper, calcium, many other elements. Calcium is not only used in bones, it's also used in muscles so that the muscles can contract. So besides the amino acid, besides just the things that we talked about before, you're going to have to throw in sulfur, phosphorus, magnesium, iron, copper, calcium, and many, many, many other elements. So when you add the extra elements to the soup, the primordial soup, the chemistry to produce a cell becomes impossibly hard. So what's the probability that these things could come together and form a living cell? The simplest theoretically possible cell would have probably about 124 proteins of about 400 amino acids each. So if the primordial soup in our lab or in our oceans contain nothing but pure amino acids, the probability that the right amino acids, those 20 amino acids, could come together to form a cell has been calculated at less than 1 in 10 to the 78,400th. What is that number? In the entire universe, it's estimated that there might be 10 to the 80th power electrons. So we're 78,320 zeros beyond the number of electrons in the universe. So if there were as many possible types of cells, there aren't, as atoms in the universe, which is about 10 to the 80th, the probability is still less than 1 in 10 to the 78,300th power. So let's have ideal conditions everywhere in the universe for trillions of years. So we put together one group of amino acids to make a cell. Wouldn't do any good. Nothing could reproduce because there would be no DNA to guide the cell's reproduction. You have to have not just a cell, but a way for it to reproduce. So what about DNA? What is DNA? It's a double helix thread, a double helix shape like the threads on a screw. One strand contains millions or billions of molecules called bases or nucleotides. They're represented by the letters A, C, G, and T, adenine, cytosine, guanine, and thymine. These can be arranged in an infinite number of ways to spell out the instructions to produce any kind of living thing. So DNA then is subdivided into chromosomes. Normal humans have 46 chromosomes unless you have some sort of a pathological condition. Other types of organisms have varying numbers of chromosomes. Then the chromosomes are subdivided into thousands of genes. Each gene contains the coded instructions either to make a specific protein or to operate some sort of a regulatory function in the cell. We used to think one gene, one protein. Well, no, genes have other functions as well. So the genes that do code for proteins are subdivided into groups of 3 called triplets. These contain the information to put one specific amino acid into place. So let's consider a very, very simple, so-called simple bacterium such as E. coli, Escherichia coli. It would take about 6 volumes of 300 pages each just to write down the instructions contained in the DNA of what's considered a very simple bacterium, such as E. coli. 300 pages, 6 volumes for a simple cell. And this is supposed to have come together at random by random chemical processes. Well, if you believe that, you can't call it science, it's faith. So it's not just that we put together the components of one cell and okay, now we're finished. No, that cell has to do something. It has to stay alive. It has to take in nutrients. It has to react to its environment. It has to excrete waste. It has to grow. So how does it do that? The parts in the so-called primordial soup, in order to come together and do something, they need catalysts. Well, a catalyst is something that speeds up a chemical reaction. This is done in many places within cells by enzymes. You probably heard of your laundry detergent with enzymes in it. Well, there are thousands of different types of enzymes, and each enzyme has a shape that allows it to snap open, snap, 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 snap open, snap close, to put together things or take things apart. So I've got just 3 examples of enzymes. This is from the U.S. government. There's a website. You can look on the screen there and see what it is. But the clamp loader complex of DNA polymerase 3 in E. coli is shown there, and then bovine chymotrypsin, and then a typical helicase enzyme. Those things are just 3 of the thousands of enzymes that are necessary to speed up chemical reactions to make living things stay living things. If you didn't have those things, then everything would be dead. It would go along just for a short time until it died, and that's it. So I mentioned enzymes. Okay, so what's the big deal about enzymes? They're special proteins called enzymes. They're crucial to the operation of cells. They act as catalysts. They make some reactions proceed billions of times faster. Without enzymes, life would be impossible. It would just go too slow and nothing would ever grow, reproduce, and so forth. So where do you get these enzymes from? DNA contains the coding instructions to make enzymes. But enzymes contains the operating ability to make DNA. So you have to have DNA to make enzymes, but you have to have enzymes to make DNA. This is one of those irreducibly complex systems. You have to have both of them together in order for it to do anything at all. So since both of these have to be present at the same time, they could not have evolved by gradual changes in dissimilar mechanisms. So you have to say, well, where would this come from? Is this something that gives indication of being a random chance process, or perhaps design? And then we're not finished. And I'm not covering every possible objection or every possible problem. I'm just doing some of the obvious major problems. Once you've got your stuff together into a primitive cell, sort of a primordial cell, I guess, in order to protect it from the environment, to protect it from being dissolved by random chemical processes, you have to put something around it to protect it. Okay, there's a layer of things, fatty compounds called phospholipids. They can spontaneously form membranes. However, these membranes don't allow many substances needed for cell growth and reproduction to get through. The phospholipids, okay, we need more phosphates for DNA in order to link together the bases or the nucleotides so we can make some DNA. So you have to have phospholipids to put around the outside of the cell some sort of a membrane, but then those phosphates are needed for further growth and they can't get in. Phosphates can't get through phospholipids. To overcome the problem, Membranes in living things contain microscopic gateways. These are called either ion channels or permeases. They let needed substances in and out of the cell. Why are these ion channels there? Because DNA contains the instructions to put them there in specific places. DNA contains the instructions necessary to put them exactly where they're going to be needed by the cell. So ion channels are needed to form DNA, but DNA is needed to form ion channels. So once again, we have an irreducibly complex system. So these are just the problems that I've covered with primordial soup. Number one, you have to have a non-oxygen atmosphere. Number two, if you've eliminated the oxygen, you have a problem where the 310 nanometer wavelength ultraviolet light is going to get through and destroy whatever you just produced. Then you have to have a sophisticated trapping mechanism so you can get out the right parts at the right time in the right place. And then you also have to be able to take the nitrogen from the atmosphere and fixate it, split it into two nitrogen atoms so it can form amine groups to go further. And then, oh, wait a minute, what are we going to do about this? We have left-handed and right-handed forms of many, many, many things in nature, yes, but in living cells, we have only left-handed amino acids. We have to eliminate all of the right-handed amino acids, which are biological garbage. And then you need to get everything to arrange itself in the right order. You have a serious problem with chemistry. And then, once we've got DNA, we could get enzymes, yes, but we can't get DNA without having enzymes in the first place. DNA is needed to produce enzymes. Enzymes are needed to produce DNA. And then your cell membrane that's going to protect the whole thing does too good of a job of protection. So, oh, sorry, can't go any further. So what if evolutionists find something that can't be explained by known natural causes? No problem. They explain it anyway. They make up stories about unknown natural causes. So what's the story? Directed panspermia. You might not recognize this term, but you've probably heard it, and it was even on an old episode of Star Trek where Kirk adopted Indian ways. Anyway, why do we have life all throughout the universe, and why is so much of it humanoid, according to Star Trek? Well, it's because there was some advanced civilization that seeded all the planets with space seeds, called directed panspermia. Sperm is a seed. Sir Francis Crick, who won the Nobel Prize for his co-discovery of the structure of DNA, he didn't do it by himself, he had a lot of other people. At least one of the ladies should have gotten the Nobel Prize. But anyway, I digress. Sir Francis Crick recognized that conditions on Earth have never been right to produce life by chemical processes. So instead of believing in creation and a creator, he proposed that some sort of seeds were sent to Earth by an advanced civilization in outer space. These seeds landed on Earth. They were alive already because the aliens sent them, and that began the process of evolution on Earth. Okay, this is very scientific, right? What's the hallmark of science? You can test it. You can observe it. You observe, repeat, test. How are you going to observe this? Not at all. This is nothing but a fairy tale. It's an attempt to get rid of God, whom you can't see, by appealing to aliens whom you also can't see. You have to believe in something you can't see. It could be a supernatural God, or it could be aliens. And the aliens don't require you to give an account for your life, where God does. So after all of these things, we've got it all together. We needed to come alive. Suppose we have two chemically identical collections of matter side by side. The first takes in nutrients. It excretes waste. It responds to its environment. It grows, it reproduces, it's alive. Well, another one right next to it does none of those things. It has perhaps exactly the same atoms in it, the same chemical composition, but it's a lifeless corpse. Why is one of them alive and one of them is dead? Science can describe how life operates, but cannot tell us what life is nor why it exists. Well, the Bible says it exists because God created it. That would be that life can only exist where God wants it to. Well, we'll talk next time about, could there be life out in outer space? Why not believe in life in outer space? Because it makes sense to you that the Bible is the Word of God, and therefore it is right in this area, which is beyond the reach of human testing. That's a religious belief, no question about it. There's a God who's responsible. Well, why believe that there is life in outer space? Because it makes sense to you that the Bible is wrong in that area. It's beyond the reach of human testing. It's every bit as religious. You can't observe, repeat, and test whether life on Earth began by divine creation or by some sort of thing from outer space. You can't test whether life out in space began as a result of divine creation or something else. Well, the Bible tells us some things that we'll get to next time. So I don't believe that there is life in outer space, and we'll go into that as we continue on the origin of life on the next episode.